Okay, hello. So this is the week 4 of the NPTEL course, Fundamentals of Protein Chemistry. And this course is coordinated by Professor Dasgupta from the Department of Chemistry, IIT Kharagpur. And I'm Bias Mukherjee, a research scholar. And in today's class, we are going to discuss the doubts that you have regarding protein isolation and purification. So I'll give you a brief overview first. So today we are going to talk about protein expression, protein purification, and we are going to talk about a lot of different techniques that are used in protein expression and purification in the laboratory. For example, affinity chromatography, size exclusion chromatography, and different other techniques. Okay, and hopefully this is going to be very useful to you because today's class is very important for your exams. Okay, so I'll try to discuss the theory first and then you can answer in the chat box. Let's see, hopefully other students will also join. And so let's begin. All right. Okay. Okay, so first thing is isolation, protein isolation and purification. So how to get proteins in pure form. Now in this class, there are a lot of things to talk about and a lot of questions for you to solve. So I will keep it short and we'll move forward very fast, okay? So isolation and purification of proteins involves separating a specific desired protein from the other cellular components to obtain it in a pure form and concentrated form to study protein structure function, and this has a lot of different applications in biotechnology, medicine, drug design, vaccine development, proteomic studies and so on. Okay, so proteins can be isolated from the natural sources. So the image on your left, on the left in the screen is showing protein rich food. Okay, and from there we are getting the pure isolated protein that is shown in the right side figure. However, to maximize the yield, they are recombinantly grown in organisms. So from natural sources, when proteins are extracted, very minute concentration is obtained. Okay, plus there are a lot of different impurities and it is very difficult to get rid of those impurities. That is why proteins are produced in a host organism through genetic modification. We'll read about all of that. That is called recombinantly grown proteins. Typically, the host organism is bacteria, yeast, insect cells, plant cells, mammalian cells, etc. using genetic engineering techniques. Now, recombinant DNA technology involves insertion of the gene of interest into the gene of interest that codes for our specific proteins with desired functions into the host organism. This is cultured to produce the protein in large scale, and biophysical techniques are employed for its extraction and purification. Okay, so we'll move on. Now, this is a diagram that explains to you how natural sources are used for the isolation of proteins. So you can see that this is a very lengthy process and there are a lot of different steps that are supposed to be involved, right? And that is why this is generally not very commonly done. And our laboratory also specializes in recombinantly expression of proteins and then characterization and then different DNA binding activities, peptide binding activities of these proteins, etc. Okay, so we do all of this on a routine day-to-day basis. Okay, so we'll move on. So now this is what is important for you. Okay, so expression and isolation of proteins from recombinant sources. So this is what we are going to talk about in today's lecture. So see, initially there is recombinant DNA technology, which tells you that there is a DNA vector. Into it the gene of interest is to be included. Now all of this is given in details for the students for whom this is new. For a lot of students this is going to be very new, and that is why everything is explained in details. Okay, but then I'm telling this to you and hopefully you'll understand. So I will not go through each and every point in details, but I'll just give you an overview. So what happens, see, the first step is cloning. So in cloning there is a vector. Okay, this vector is, it can be a cloning vector, it can be an expression vector. Let's say that there is a naturally found protein and you want to introduce some mutations in it. Okay, so we'll talk about cloning and expression vectors in the upcoming slides. Now just remember that for any genetic modification and for ensuring that this mutation is persistent, it is staying there in the vector and this bacteria is preserved for a long period of time, for that cloning vectors are used. But then for expression vector, this vector is engineered in such a way that it is made so that large amount of protein can be expressed in the bacteria. So that is your expression vector. Okay, so what happens is first cloning of the gene of interest into the vector happens. Then it is transferred into this expression system. So for example, PET28A is a very commonly used expression vector, and then this is inserted into the bacteria through let's say transformation, or it is inserted into the mammalian cell through transfection, and then it is grown. Okay, and then after that, from that bacteria, the bacteria is broken open using, let's say, sonication. So the cell wall is ruptured and all the contents are taken out, and then from those contents our specific protein of interest is isolated. That is the full procedure. Okay, now see what happens. So this is the entire process that is shown. So first is expression in E. coli. So after cloning and after expression, after cloning, this is transformed into the bacteria and now expression in E. coli happens. So normally for protein expression, E. coli strains are used. So normally BL21DE3 is the strain that is used. Okay. Now see, inoculation of the media with the transformed cell culture. So basically there is this Ludia Bertini broth, and this is made previously and it is autoclaved to make it completely sterile. Now this broth is like you just add water and then this is ready. Okay, and what is it made up of? It is made up of yeast extract, it is made up of tryptone and everything. So all of the ingredients can be separately added to make LB. Otherwise, prepared LB is also purchasable from the market. Okay, now we just add in water, we autoclave it and we keep it, and it is completely sterile. Now from our bacterial culture that we have stored in -80-degree refrigerator, or you can also take in from some colonies from a plate. From there you take in the culture and you dip it into this bacteria, into the LB, and then this is grown for a particular amount of time depending upon how much time it takes for that protein to express. Okay, so initially it is overnight, it is grown, and then after that IPTG is introduced, and then let's say a particular protein requires 5 hours for growth, or maybe it requires 3 hours for growth. So that has to be optimized, plus the temperature at which it will grow, that also has to be optimized for different proteins. Okay, so what happens is this culture that in which the bacteria has grown, let's say you keep it for 5 hours and then it has grown, then what you do is you centrifuge it, and then all the media is there on the top and this is there as a supernatant, and all the pellet contains your bacteria. Next what you do is you throw away the supernatant and you preserve the pellet in a particular buffer. Okay, and you resuspend this pellet into a buffer and you store it. Then what you do is you sonicate it. So see, this is the soluble lysate. This is called the cell lysate, and then you sonicate it. That is, you pass ultrasonic waves into this cells, bacterial cells, so that the cells rupture, and all the components in the cell come out into the media. Then what you do is you centrifuge once again, and now all the proteins, these come into the soluble fraction, that is the supernatant, and all the bacterial debris, the cell wall and all the parts of the bacteria that are broken. Okay, so all of that goes into the pellet. So now what you do is you throw away the pellet, you take the supernatant, and this supernatant is taken for different chromatography. So let's say we will talk about this in the upcoming slides. Okay, for example, there is this histidine tag in your protein. So 6 histidine residues are added at the beginning of the protein, or maybe at the end of the protein, depending upon your requirement. Okay, so the vector that I talked about, PET28A, this vector has this histidine stretch before the protein at its end terminal, already integrated inside the vector. Okay, so and if you're using PET28B, then this histidine stretch will be at the end of the protein, so C terminal of the protein. Okay, so now what you do is you take this, and let's say you are doing, if this histidine stretch is there, or let's say there is GST, there is some kind of affinity tag, then we will go for affinity chromatography. We'll talk about this in the upcoming slides. Okay, so using this chromatographic technique, you separate out the protein, and then what you do is you go for, let's say, size exclusion chromatography for further purification, and then you go for dialysis. So dialysis means it is there in a particular buffer and you want to change it into the buffer of your requirement at which your protein is active and it is functional. So you change it into this particular buffer, and then you go for the different assays. Okay, and then also in between, using size exclusion, using page, SDS page, you check whether your protein is there or not, whether it is having any impurities or not. If there will be impurities, there will be other bands also other than your protein. Okay, so that is the entire process. So this entire process takes around 2 to 3 days minimum. Okay, that is minimum, right? So now we go for the details of it. So the first one is cloning and expression vector. So see, this cloning vector is used to acquire multiple copies of the foreign DNA fragment, that is gene of interest, and it has a very high copy number. Okay, and they have features such as this ORI, that is the origin of replication. It is self-replicating, restriction sites, etc. And it is also having the gene of resistance. Okay, so you're growing it in, let's say, kanamycin containing media, then the vector will have kanamycin resistance gene inside it. Okay, so that the bacteria is forced to retain this particular vector within it and it does not throw it out. Okay, now for expression vector, it's very important because see, this contains this ribosome binding site, because we need translation, only then the protein is going to be formed, right? So that is why ribosome binding site is there, promoter sequence is there, then there is start codon, there is stop codon, there are enhancer sequences, all of these termination sequence, initiation, etc. All of these different sites need to be there in the vector. So diagrammatically this is explained for you. Okay, you can go through this. Now, what are these host cells? Now normally, commonly I was talking about bacteria, because doing it in bacteria is relatively very common. A lot of people grow proteins in bacteria. But what if your protein requires some kind of post-translational modification? Or what if the protein size is very large? Then bacteria will not be able to synthesize it, right? So for that, there are different kinds of host cells. For example, there can be these mammalian cells, there can be insect cells, there can be E. coli cells or bacterial cells, there can be other bacterial strains also, like for example tuberculosis, Mycobacterium tuberculosis strain, and these different other strains. Then there can be yeast. So there are different properties of these different host cells. So see, for example, CHO cells and HEC293 cells, these are useful if you want to incorporate post-translational modifications. And also, but then the culture duration is very long and it is very expensive. So there is advantage and disadvantage. Both are mentioned there. And then insect cells are also useful for post-translational modification, and partial glycosylation is also possible here. Now for E. coli, this is the cheapest and the fastest. Okay. And for yeast cells, only some post-translational modifications are allowed, others are not, and this is also relatively cheaper compared to the mammalian cell culture and others. Okay. Now, so see, the entire process that we have talked about, that is again explained here. So the first step is gene cloning. This you already have understood by now. Okay, then there is transformation. So transformation, you're going to insert the DNA, which is made up of vector plus your DNA of interest into the host cell. That is transformation. If the transformant, if the it is not bacteria, if the host cell is not bacteria, it is a mammalian cell or an animal cell, then it is called transfection. Okay, now see, cultivation and induction. So what happens is when you're growing it, so there are different kinds of cultures. So first is primary culture. So primary culture means that you want the cell density to rise up, and then after that you want to start with the protein expression, because initially when the bacteria is there, it needs some time to adjust to the surroundings, right? So you're supposed to give it some time to adjust to the surroundings. So this initial lag phase goes on. Then there is log phase, and when the bacteria is at the log phase or at the saturation phase, you take it out and then you go for secondary culture. That is in the big flask image that you saw earlier. Okay, so growth of the transformed bacterial cell in a suitable growth media. And then once the bacteria has reached an optical density of around 0.6, so that means there is enough population of bacteria in the culture already, then you want to introduce IPTG. So what is IPTG? IPTG is actually a homologue of allolactose. Now you might have already studied about operons in bacteria, right? So we will discuss all of that in details in the upcoming slide. So IPTG is something that tells the bacteria, now is your time to start synthesizing this protein of interest. Now why is that so? Because if let's say that the bacteria starts synthesizing your protein of interest directly, right from the very beginning, then what will happen is the proteins can be toxic for the bacteria and the bacteria will die eventually. So a lot of protein will not be synthesized. Before that, the bacteria will be dead. Why is that so? Because the protein of interest is not a natural thing that is required for the bacterial growth, right? It is utilizing the machinery inside the bacteria to prepare something that is not even necessary for the bacteria. And this is often even toxic for the bacteria. It in fact kills the bacteria when it gets accumulated in a large concentration. Okay, so we'll read about that very soon. So after that, see, cell harvesting. Cell harvesting means that you're spinning it so that all the bacterial cells come in as pellet and the supernatant is there, which is just media, and you're throwing it out. Okay, so after a specific incubation period, which is around 5 hours or more, when the protein of interest is expressed at high levels, resuspend the pellet containing the bacterial cells in lysis or wash buffer with a protease inhibitor. For example, PMSF and sodium azide to prevent fungal growth. So PMSF is important because see, this is actually a phenyl methyl sulfonyl fluoride. So this is useful because it prevents the serine proteases from cleaving off the proteins. And IPTG is a molecular mimic of allolactose, and this is synthetic. That means the bacteria cannot use it up. It cannot metabolize it, right? So non-metabolizable. That means once you add it into the media, it is going to be there in the media at all times, and you don't need to add it again and again. If that was allolactose, you would have had to add it once again and again, right? Because the bacteria would metabolize it. Now it triggers transcription of the lac operon and therefore is used to induce protein expression where the gene is under the control of the lac operon. Okay. Now we were talking about these BL21 DE3 cells, which are the most commonly used cells for protein expression. So what happens in this BL21 DE3, and what is DE3 after all? So see, in a BL21 cell that is normally used for protein expression in the laboratory, the cell is there inside the genomic material of the bacteria. A part of this lambda D3 phage genome, which is a bacteriophage, that has been taken. It's a viral genome. Okay, so that has been taken and introduced into the genetic material of the bacteria. This is the D3 part. And this D3 part contains this mutated lac operon. So this lac operon has the it is under the control of this lac operon, and the gene, the structural genes that are there in the lac operon, this is removed. Okay, so now what is an operon? Operon means that a lot of different genes are there and they are going to function for a specific cause. So they are under the same regulatory mechanism. That is an operon. A set of genes are there and they are under the same regulatory mechanism. Okay. So lac operon is a very simple kind of operon. So there is this lac operator and there is this lac promoter. So this lac I is the lac inhibitor gene. When this gene is active, it is producing a protein which is called the lac repressor, and this repressor is going and binding to the operator site. Now, if this is the promoter sequence, this is the operator sequence. So when the operator sequence is blocked, then the T7 RNA polymerase, sorry, E. coli RNA polymerase, this is not able to come and bind to the promoter. If it is not able to bind to the promoter, then the structural gene is not expressing. That is the gene in front of the lac operon. This is not expressing. And this structural gene is actually replaced, and there the T7 promoter gene is there. Now T7 promoter gene means this is a gene which codes for a particular enzyme which is called the T7 polymerase, T7 RNA polymerase. T7 is once again a bacteriophage, and this T7 RNA polymerase is 5 to 10 times faster and 20 times less error prone compared to a E. coli RNA polymerase. That is why T7 RNA polymerase is used for bacterial overexpression of our required proteins, our protein of interest, right? So see, normally what happens is this lac repressor is preventing the production of this T7 gene. But when you're adding IPTG in the media, that time this T7 gene is expressing, because the lac repressor is no more able to bind to the lac operator, and therefore E. coli RNA polymerase is coming and binding to the promoter region, and that is why T7 RNA polymerase is expressing. This T7 RNA polymerase goes and in your PET, that is the vector that you are introducing, there is a promoter region for this T7 RNA polymerase. So T7 RNA polymerase gets synthesized and it comes and recognizes its promoter region and binds to the promoter region, and the downstream gene is your gene of interest. It's the one that codes for your protein of interest, right? So this is your target gene. Now what happens is so this is the entire mechanism of BL21DE3 expression system. Now something interesting is there which is called BL21DE3 P lys S. So in P lys S, there is one more very tiny system, plasmid system, where actually it codes for a particular protein which inhibits the activity of this T7 RNA polymerase. Now, lac operon is a leaky operon. That means even when IPTG is not added, very minute amount of T7 RNA polymerase can come and bind to the promoter, and small amount of this T7 RNA polymerase can be produced. Okay, so even small amounts of T7 RNA polymerase, if it is produced, it will come and make your gene of interest, make the protein of interest, and that is why once again the bacteria can die. So when very small amount of T7 RNA polymerase keeps on getting produced, that time this T lys gene, this forms this T7 lysozyme which prevents the activity of this T7 RNA polymerase. That is why your gene of interest is only going to overexpress when IPTG is added. Okay, but when IPTG is added, T7 RNA polymerase is produced in such large excess that this inactivating protein is no more able to control it. Okay, that is how it becomes active. Right, so we have talked a lot. Now you can try to answer this question. Okay, it's a simple question. You can answer in the chat box. Why are some recombinant proteins toxic to the host cell during overexpression? So the first option is they destabilize the host genome. The second option is they compete for tRNA and ribosome, impairing the host translation. The third option is they interfere with essential host pathways or form aggregates that burden the cell, and the fourth option is all of the above. So why do you think our protein of interest can be toxic for the host cell in which it is growing? Why do you think that is the case? You can answer in the chat box. Okay. Okay, we'll look at the answer then. Okay. So the right answer is D. So recombinant proteins are toxic during overexpression because they impose a high metabolic burden on the host cell, leading to protein misfolding, aggregation into inclusion bodies, disrupted cellular processes, and triggering cellular stress response that inhibit growth and viability. Right. Additionally, some proteins are intrinsically toxic if they perform a disruptive or destructive function within the host, such as cleaving the host mRNA, or if the host lacks the necessary machinery for proper folding and post-translational modification. So these are the reasons our protein of interest can be detrimental to the host bacteria in which you're growing it. Okay. Now, so this is the entire workflow of the protein expression and purification process. Right. So initially there is protein production, and for protein expression you have to first select the bacterial or any host organism strain that you want. You have to go for codon optimization. So codon optimization means that, let's say, you know that one particular amino acid can be coded by, let's say, 2-3 codons. Okay, so they are degenerate. Now in a bacteria, in that particular strain of bacteria, let's say that there are 5 tRNA available for one codon and 2 tRNA available for the next codon and only 1 tRNA available for the third codon. So for that particular amino acid, then you have to select the codon with the maximum number of amino acids, maximum number of tRNA that have the corresponding anticodon. Okay, so that is how you optimize the codon for better protein expression. Next is fusion system. So fusion system means you make one protein join with your protein of interest, and this protein can be a GST tag. So it's a huge protein, it is highly soluble, and it makes sure that if your protein is not very soluble, then it will make sure that this protein also becomes soluble. Okay, and this is also useful in affinity chromatography. Okay, so glutathione S transferase, that is your GST. Okay, co-expression. Co-expression, you can express this protein with another protein, and mutagenesis, you can introduce mutations into the protein, and isotope labeling. So let's say you're making samples for NMR spectroscopy. Then what you will do is, instead of normal nitrogen, you can introduce N15. Instead of normal carbon, you can introduce carbon 14, etc. Right? Protein purification. So the protein needs to be soluble. If the protein is not soluble, you have to make it soluble. You have to introduce mutations and other ways for solubilizing the protein. Then we come to membrane proteins, which are even difficult to work with. Right, so this is how different things are done. Characterization, so once the protein is obtained, once it has been purified, you can go for mass spectrometry, you can go for biochemical and protein interactions, etc. Structure solvation, etc. These can be done. Okay, this depends upon what you want to do with your protein of interest. Right, so now we have one more question. So we talked about proteins becoming insoluble, right? So see, recombinant proteins often accumulate in the pellet fraction after cell lysis. So cell lysis means, I told you about sonication, right? You're introducing ultrasonic waves, and because of that constant vibration, the cell wall is rupturing. And once the cell wall ruptures, the proteins, they are normally what? Water soluble. So the protein should come, when you spin it in centrifuge, the protein should come into the supernatant, and all the cell debris, broken pieces of the cell, this should go into the pellet. But sometimes that does not happen. Sometimes even the protein of interest gets deposited as a white big white layer on the pellet, and that is a very sad thing to see because you can't work with it anymore if you don't want to go for inclusion body wash and this entire other big process of refolding it. Okay, so why do you think that happens? Option A is they undergo glycosylation and become insoluble. They form inclusion bodies due to misfolding and hydrophobic aggregation. Third option is they are degraded by host proteases into insoluble fragments. Fourth option is they bind tightly to the host DNA and RNA. So which one do you think is happening in this case? You can answer in the chat box. Okay, try answering. Let's see how much you people are understanding. Nobody is stopping me. Nobody is asking me any questions. I'm surprised. What is going on? Are you people still understanding anything? Or do you understand everything? That's why you don't have any questions. Try to answer this question. You can at least make a try. Otherwise, we can look at the answer. Okay. Since no one is answering, we'll check the answer. So see, the answer is actually B. So these proteins, when they are produced in excess, what happens is some proteins, they have these intrinsically disordered regions, right? So there is a folded region and there are these N and C terminal regions which are very, very flexible. These are not very well folded, right? These are forming these random coils. So these intrinsically disordered regions, and let's say a protein has a lot of cysteine residues, then it is supposed to form disulfide linkage, but sometimes it forms wrong disulfide linkages, and then there is a high chance of misfolding. So when that happens, all the proteins, these, when there is, let's say, aggregation. So let's say that a protein has a property of a lot of, it is produced in excess, right? And the protein comes and aggregates, and it, through hydrophobic aggregation and misfolding, all the protein goes into this inclusion bodies which are there in the cell, and these are these can be like you cannot, so the inclusion, when the protein goes into the inclusion body and you spin after sonication, you will see that this is the part where they will go into the pellet. And so for that there is a different procedure which is inclusion body wash, and from there you can retrieve your protein back, and the material from where you can study about this is given at the bottom of the page. This is the DOI, so that you can refer to for knowing more about this. Okay, right. So we have two more questions here. Try to answer these two questions. Okay. So the first question tells you, to recover proteins from inclusion bodies, the most commonly used method is, one is high salt precipitation followed by dialysis, solubilization with cheotropic agents such as urea, guanidine hydrochloride, followed by stepwise refolding, and C is using protease inhibitors during lysis and expressing the protein at higher temperature to increase folding efficiency. Okay, so which one do you think would be the right answer? So the protein is in the inclusion body, that means there is a lot of aggregation. So what you can do is one of these four things. Okay, now for the second question, that is question number 7 that we have on this slide. So lowering the expression temperature in bacterial system often improves recombinant protein solubility. This is because A is translation rate decreases, allowing the protein more time to fold properly. Proteases are less active because proteases are most active at 37 degrees centigrade. That is the normal temperature for the bacteria. But let's say you are growing your protein at 16 degrees centigrade, then the proteases will not be very active, right? And then hydrophobic interactions are weaker at lower temperature. And D is all of the above. Okay. Right. People are getting discouraged and they're leaving. We will look at the answers. So the answers are second option for question number 3 and fourth option for this question number 7. So see, what happens is when the protein is going into inclusion body, then high salt precipitation followed by dialysis is not going to help, but then you can actually solubilize the protein during this sonication step. You can add in urea. You can add guanidine hydrochloride, which is also similar to urea. You can see the chemical formula, it is very similar, right? And these are actually agents that help in further unfolding the protein. So when you're completely unfolding the protein, then all the interactions of that particular protein with other proteins, nucleic acids, and all everything else is gone, right? So the protein is just a linear chain now. And this, if it is a linear chain, it will be made up of these amino acids which are charged. So it will definitely interact with water and it will definitely become more soluble. So now when you spin it, all the protein, your protein of interest will go into the soluble fraction. So that is how you can solubilize the protein. And then when you are passing it through this affinity chromatography, it will refold into its functional native state, and this is called on-column refolding. So when you're passing it through the column, it will refold. Now we in our lab use this a lot. Why? Because these proteins are proteins, the ones that we work with. Now these are DNA binding proteins. So even after we are adding DNAs is chopping off all the DNA that the protein is bound to, right? So these are transcription factors. Now the problem is that there will be small bits of DNA that will be still attached to your protein, because the protein binds to 4 to 6 base pairs of DNA. Now these small proteins, small DNA fragments, this will be bound to your protein. So later on, if you want to, after purification, if you want to go for any DNA binding studies, you will not get good results because this DNA is already bound. So now when you are adding DNA externally, the protein is not binding properly to this DNA, right? So that is why it is very important that you are adding these denaturant. When you are adding denaturant, it is completely removing all interactions. The protein is becoming one linear chain and then you can refold it. So that is one way of doing it. Another way of doing it is IB wash, which is inclusion body wash. So this inclusion body is subjected to a lot of different buffers and then it is refolded. Okay. Now for the next question. The right answer is all of the above. So what happens is sometimes when the bacteria is triggered to produce proteins at 37 degrees centigrade, it produces so much of protein that all the protein is either misfolded or it is aggregated and it is completely denatured and it cannot be used anymore for further studies because it goes into this inclusion body. Now to prevent that, what you can do is you can grow it at a lower temperature. Let's say 30 degrees centigrade or 22 degrees centigrade, 16 degrees centigrade. Then what happens? Translation is definitely decreased because all the enzymes, everything works best at 37, right? So proteases are also less active, so they are not able to chop off your protein. Hydrophobic interactions are weaker. So all of these points are correct, and so D is the right option. Okay. Right. So these are very practical questions. Now I have included these questions because everybody is studying from the textbooks. Right? You people have already studied so much from textbooks. You will anyway get bored. That is why when you look at these real life practical problems, you will have an idea that okay, this is how we can actually troubleshoot if something goes wrong. So these are important troubleshooting steps and these are also important questions which you can expect in exams. Okay. Right. Now we will look at something which is very important for protein purification, and this is the concept of precipitation. Now what is salting in and salting out? So see, proteins are very delicate and they are best soluble at only a specific temperature and pH range. Beyond that, they are less soluble. They actually precipitate out, right? So what is salting in? So at low concentration, added salt usually increases the solubility of the charged macromolecules because the salt screens out the charge interactions. And so low salt prevents aggregation and therefore precipitation. So this is called salting in. So little amount of salt is in fact helping in protein solubility. But then when you add too much salt, then what happens? At high concentrations, added salt lowers the solubility of the macromolecules because this added salt actually competes with the protein to interact with water. And of course, salt is going to interact better with water because it is very small and very strongly charged and has very small, tiny molecules, right? And that is why the solvent interaction is improved and the protein protein interaction increases. That is why aggregation and precipitation happens. Earlier in salting in, it was protein and solvent interaction that was increasing because of the addition of salt. Okay, so high salt removes the solvation sphere from the protein molecules and they come out of the solution. So solvation sphere means that this is your protein, it is surrounded by water molecules. Right? When salt comes in, the salt is going to interact more with the water molecules. When there is too much salt, then what happens? Protein is going to interact more with other proteins and entire thing is going to come out as a precipitate. So this is called the precipitation line and this is your solubilation line and the entire figure is explained. Now coming to salts, some salts are very useful in helping in precipitation. Some salts are very useful in helping in salting in. Okay, for example, ammonia. Ammonia is very useful in making your protein soluble. So there is this entire list which has been arranged based on the ability of salts, ability of ions to solubilize proteins or precipitate proteins. This is called the Hofmeister series. So Hofmeister series is the classification of ions based on their effect on the solubility of protein as well as on the stability of secondary and tertiary structure of it. So choose the correct order of salts for salting out process, right? So it is given here. So salting out. For salting out, which order do you think would be the right one? Today nobody is answering. Okay, we'll look at the answer. Right. So see, this is your Hofmeister series. It's given this way. Okay. And the salts that decrease protein solubility are, one second. So see, so the salts that are increasing surface tension of water and decreasing protein solubility, decreasing protein denaturation and increasing protein stability and increasing protein hydrophobicity, these are called cosmotropes. And the others that are called chaeotropes. So chaeotropes are increasing protein solubility. So chaeotrope is urea, guanidine hydrochloride. These are denaturing the protein and that is why increasing the solubility of the protein. But they are also decreasing the stability of the protein, right? Because the secondary and tertiary structures are broken. So that is how you will remember. So chaeotropes are causing chaos in the protein solution and opening up the complete protein, in turn solubilizing the protein. Cosmotropes are actually maintaining the stability of the protein and these are weak, very improving the protein hydrophobicity. Okay. Now, so during purification of a membrane protein, detergents are often added. That is because A, they precipitate the protein, making it easier to separate. They mimic the lipid bilayer, solubilizing the protein in aqueous solution. C is they inhibit proteases that degrade the protein. And D, they denature the protein, aiding refolding. Now here, I have added the questions first and then the concepts later so that it helps you to draw your attention first, right? So if I talk about the concepts first, you will obviously get bored. That is why first the question and then the concepts. Okay. So detergents are very useful for protein purification because they mimic the lipid bilayer, solubilizing the protein in aqueous solution. Okay. Now, protein separation techniques are often based on the following. Like, there are a lot of different proteins in a solution and I want to isolate my protein of interest. So, which things am I going to target? First is solubility of the protein. Second is viscosity of the protein. Third is charge of the protein. And fourth is specific binding of my protein. Now, these three things, there are three, I'm giving you a clue there. So, the properties that we are talking about here, so these properties are exploited by protein purification techniques, certain chromatography techniques. Right? So, once you are able to answer this, I will tell you about the different chromatographic techniques. So, solubility of the protein, viscosity of the protein, charge of the protein, and specific binding of the protein. So, which one do you think is the right answer? C. Yes, that's right. Yes, very good. Finally, you're answering. Okay, good. So, see, solubility of the protein. All these things that we studied about, precipitation, salting in, salting out, all of this are exploiting the solubility of the protein. Next is charge of the protein. So, anion exchange chromatography exploits anion exchange and cation exchange chromatography exploits this charge of the protein. Next is specific binding of the protein. So I talked about histag, I talked about GST fusion proteins, right? So these are exploited by the affinity chromatography. Okay. Now, these are the different types of chromatography techniques that exploit different properties in proteins. So, affinity chromatography, specific binding interactions, ion exchange, charge of the protein. Size exclusion chromatography, molecular size. Hydrophobic interaction chromatography, hydrophobicity comes from the name, right? And gel electrophoresis, charge and size. Now, you have already studied about gel electrophoresis. Then comes isoelectric focusing. So this depends upon the pH of the protein, at which pH the protein is going to precipitate, right? And then there is precipitation. So salting and salting out, solubility changes. And liquid-liquid extraction. So this is partition coefficient. Right. Now see, there are some questions on enzymes. Now the thing is that these should have been a different class. This should have been kept for a different class. But since you have questions from enzyme activity and enzyme specificity in the previous year's questions, that is why I have included this part also. But then this is a very diverse topic, and so we can go through this quickly, but then you will have to give a lot more time to this. Okay, so during a successful purification scheme, it may be expected that specific activity increases, specific activity decreases, the number of proteins in the sample decreases. So what happens in purification? Okay, so from a lot of proteins, you are isolating your protein of interest, and what can you expect from there? Let's say that your protein of interest is an enzyme. So what is going to happen to the specific activity and number of proteins in the sample? A. Okay, let's see. So specific activity is enzyme activity divided by amount of enzyme. Okay, and yes, that's correct. A is the right answer. Okay, so specific activity increases and the number of proteins in the sample decreases because your protein is only being taken out. Rest of the proteins are getting removed, right? Okay. And since the amount of enzyme is decreasing, then definitely the specific activity is going to increase. Okay. Now see, The number of molecules in the isolated substance must increase during purification, and specific activity of a protein must increase as the purification progresses. And the definition is given here, the one that you saw in the formula. Next important thing is you need to remember all of this thoroughly, because there will be questions on enzyme activity, and I am not going into the details of this because all the slides will be available to you, so you can go through all of this in details. Okay. So see classical units and international units, and there will be numericals. There will be this numerical aptitude test which will ask you to correlate between these classical units and international units. Okay, enzyme activity, specific activity, molecular activity, all the definition and the formula you have to remember, because there will be questions where all of this is going to be asked. Okay, so all the formula, all the different formula that is required for this particular enzymes topic is given here. All the formulae are listed here. So please go through this, and hopefully this is going to help you to remember things. Now, since we have not discussed this in details, I will skip this enzyme question and you can go through this later. Okay. So see recovery of enzyme activity and cold purification. So everything is explained here, over here in the previous slides, and you can refer to all of these slides because we have a lot to cover today. That is why you guys can read about this, okay, once the slides are available to you. Right. So we will start with ion exchange chromatography. Now hopefully this is going to be very interesting for you people. Okay. So what happens in ion exchange chromatography is that this is based on reversible electrostatic interaction between charged biomolecules, which are the analytes, and oppositely charged functional groups on the stationary matrix, which is called the resin. Okay. Now elution is carried out by increased concentration of the counter ions. So what are counter ions? Let's say for anion exchange, your protein is an anion, it's negatively charged, and the resin to which it is going to bind, this is positively charged, right? And the other ions which are also negatively charged but they are smaller in size compared to your protein, these are called the counter ions. So counter ions are going to come and bind to the resin, displacing your protein of interest, and this is going to get eluted, right? So this is what is the principle. Now see the anion present in the sample displaces the mobile counter ions, and anion exchange resin is there which is normally positively charged. Now see for cation exchange, carboxymethyl cellulose is used as the resin. Now carboxymethyl, so this is negatively charged resin, and because the carboxy group is there, that is why it is negatively charged. And the buffer, this buffer is normally 4 to 7. It's a pH of around 4 to 7. Why? Because it's a cation exchanger, right? So when the PI is such that the pH is kept at this range, then the protein is going to have positively charged. Okay, so in the first class we talked about amino acids, we talked about the PI and the pH, right? So you can refer to that. And let's say there is an anion exchanger. So in anion exchanger, the resin is positive and the protein is anion. So DEAE cellulose is used, and the buffer pH is around 7 to 10 is the buffer pH. Okay. Now see, we will look at this in details. So see, there is a negatively charged anion and this is binding to the positively charged resin, and initially when your protein is negatively charged but the rest of the bacterial proteins, these are all positively charged, then initially in the flow through, you are going to get the positively charged other proteins. Then you are going to introduce this high salt buffer, which is going to contain the negatively charged small ions, and these are going to come and bind to the resin, and it is going to elute your protein of interest. Right? So that is what is going to happen. The paper is given here as reference. You can go through it. Okay. Now we will go for affinity chromatography. So what happens in affinity chromatography? So one thing that I can ask you is, let's say for a very highly, let's say for a protein which is highly basic. So its PI is going to be very high, right? And so for that protein, if the pH is around 7, let's say that the PI is around 9, then what is going to be the charge of this protein? Positive or negative? Which one do you think? You can answer in the chat box. The protein has a PI of around, let's say, 11. Okay, its PI is 11. It has a lot of basic residues. Now, the pH of the solution is kept 7. So what is going to be the charge? Whether it is going to be positively charged, whether it is going to be negatively charged. Based on that, you will have to decide whether you're going to use a cation exchanger or an anion exchange chromatography. Right? So that is how you're going to do it. Okay. Now we'll carry on with affinity chromatography. Now see, here they are going to exploit the property of attraction between different, right? Okay, it's going to have negative charge. Is it? Look up. Okay. Let's see. We'll find about that in the upcoming questions. It's going to be clear. Right. So let's try to understand affinity chromatography first. Okay. So in affinity chromatography, so interactions, different interactions are going to be exploited. Okay. For example, antigen-antibody interaction, enzyme-substrate interaction, then protein-ligand interaction, receptor-ligand interaction. Now lectin. Lectin is a protein which binds to carbohydrates. Okay. Its interaction with carbohydrate. Then nucleic acid and complementary base interaction. So all of these different things, these are taken into consideration. And metal ions and polyhistidine fusion proteins. So now I was talking about this histidine tag in the beginning of the class, right? Now the 6 histidine residues, they have the property, the imidazole ring in the histidine has the property to bind to this metal ion. For example, nickel or cobalt, right? So this is one of the properties that are exploited here. Then we talked about GST, which is glutathione S transferase. This binds with glutathione, which is represented as GSH. Okay, so now the most commonly used purification tag is histidine tag. So in this histidine has a property of binding to nickel. Now how will you make sure that there is nickel in the column? So there to the agarose bead a particular chemical entity is attached. This entity is called NTA, which is nitrilo-triacetic acid. Now this NTA binds to nickel through conjugation complex, and the imidazole ring in the histidine of protein binds to this nickel as well. Right, so this entire thing is called immobilized metal affinity chromatography or IMAC. Now this nickel, once the protein of interest binds to nickel and then you wash it, the protein of interest is removed. Right, later on when you recharge the column, you remove all the bound proteins using NaOH. Then you wash. Then you add EDTA, and EDTA is a chelating agent, so it chelates all the nickel and it removes all the nickel. Then you once again add NiSO4 solution, and the nickel goes and binds to the column, and it is ready for protein purification, right? So this entire thing is nickel is a covalently coupled. It is covalently coupled using NTA to agarose, which is a hexadentate ligand that coordinates nickel ion and histidine residues in the amylite. Naturally found. Now histidine stretch can be there in a protein naturally or recombinantly synthesized, that is using histidine 6 tag, binds to this coordination complex. Normally nickel and cobalt are the metals used, and FPLC exploits Ni-NTA affinity chromatographic technique to purify histidine 6 tagged proteins. FPLC means fast protein liquid chromatography, that is its full name, and elution is either biospecific. So biospecific means that you can use free imidazole molecules in the elution buffer. So you know that imidazole binds to nickel, and when you're adding free imidazole rings, it is more mobile, it is smaller, it is going to easily come and displace your protein of interest and bind to the nickel. Or it can be non-specific, that is increasing concentration of salt. So that is going to weaken the Coulombic interactions, and it is going to weaken the other interactions and make sure that the protein is eluted. Okay. Right. Now, that was all about your affinity chromatography. So similar to nickel histidine tag, there can be all of these other interactions. For example, there can be GST and GSH interaction. These other flag tag and all of these different other tags are there. You can read about those. Right. Now we come to size exclusion chromatography. So in size exclusion chromatography, what happens is there are pores of different sizes throughout your column. Now you pour in the proteins. Now the proteins that is the largest in size, it will not enter into any pore. It will be too large for the pore. So it will come out first. Next, when the medium sized proteins are there, these will enter into some pores and then elute later. And the smallest ones, these are small enough to enter into all the pores. So it is going to enter into all pores and it is going to come out last. Okay, and from there, this is how you are getting. So large molecules, then the medium size, then the smallest. And the partition coefficient gives the affinity of the compound for the stationary phase. Okay, so this is the formula that is there, and all the different terms are explained in the next slide. So see, this is what the formula shows. So Vt is the total volume, Ve is the elution volume, that is the volume at which the molecule of interest elutes. Volume means you're constantly passing in buffer, right? And so assuming that it is within the fractionation range of the matrix. Now V0 is the volume of the space between the beads. For example, if these are the different beads, there will be space in between beads. This is called the void volume. Okay, and Vi is the volume of the space within the beads, V internal. So if you use all of these different values, you can calculate the partition coefficient. Now applications of size exclusion or gel filtration chromatography is that it is very effective in determination of molecular weight of a particular protein. It is useful for getting the different fractions of a protein. You can even learn if a particular protein exists in different conformations. For example, one conformation has a different size, then it will elute first, and then there will be this overlapping peak, and another conformation of the molecule will maybe elute if it has a different size and different folding. Okay, buffer exchange is also done using size exclusion chromatography. Right. Now another important thing is HPLC, high pressure liquid chromatography. Now this is very important but very expensive, and it's also very effective. Okay, so the separation in the most effective one is reverse phase HPLC. So in reverse phase, what happens is initially there is the stationary phase and a particular molecule of interest. This is binding to the stationary phase based upon the hydrophobic interactions. Then it is reversed, and when it is reversed, it is eluted out. Okay, so the separation in the reverse phase HPLC is based on hydrophobic interaction between the non-polar analyte in the relatively polar mobile phase and non-polar stationary phase. So stationary phase is non-polar and your molecule of interest is also non-polar, but the rest of the mobile phase is polar. So these two bind together due to hydrophobic interactions. Now analyte with higher hydrophobicity will interact more strongly with the stationary phase and will take longer time to elute from the column. This desired analyte is eluted by gradient elution, that is relatively increasing the hydrophobicity of the mobile phase. Right, so when the composition of the mobile phase is changed during the analysis. So application is separation of nucleic acids and other biomolecules with very high efficiency. This is in fact used for purification of vaccines, mRNA based vaccines and other vaccines as well. So it's very effective. Right. Now SDS page. SDS page you guys have already studied. What is a native page? A native page, the normal native, the protein is in its native naturally active form. It is binding to another protein or a nucleic acid and you can visualize it. And in normal SDS page, the protein is completely denatured. It is coated with SDS and it is running on the gel based on the difference in molecular weight. So we have studied about this in the previous class. Correct? Okay. Now you have to answer this particular question. So you know that a protein that you want to purify from a natural source forms a multimer. Now these are the kind of questions that you can expect in your part C in the CSIR exams. Okay, so these kind of questions make you think, and there will be questions that will ensure that you know about a lot of different techniques and you know how to answer these questions based on that concept. Okay, so these kind of questions are the ones that are very important and you should be able to solve these. Practice more of these kind of questions. So see, you know that the protein you want to purify from a natural source forms a multimer with multiple subunits giving a molecular weight in solution much bigger than visualized on the SDS page. In SDS page, all the components break apart and you get only the size of the monomer, right? There is only a small amount of target protein in the total sample of which of the following is appropriate purification strategy: affinity chromatography, ion exchange chromatography and size exclusion chromatography. So how are you going to separate this out? It forms a multimer. Okay. C is yes. Okay, right. Okay. So see that the molecular weight of the protein in solution suggests that ACC would be appropriate. However, the filtration properties of ACC matrices compared to capture properties of IEX or affinity chromatography matrices means that only small amount of sample volumes can be loaded onto ACC columns, typically 1% of the column volume. As your target protein is already quite dilute, an unreasonably large column would be necessary for purification of a useful amount of protein. ACC also dilutes samples further during chromatography. A capture step such as ion exchange chromatography, affinity chromatography or hydrophobic chromatography would be a good step to concentrate the protein while also removing the impurities. As the protein is to be purified from a natural source, the affinity chromatography matrices used for recombinant proteins will not be suitable because there would be more unusual affinity matrices specific for your proteins. IEX is a good capture step. Adding a ACC gives you additional purification using different property of the protein. Okay, so based on this, the first one would be more appropriate. Okay, now we move on to something called differential centrifugation. Now you guys might have already read about this. So what happens is when you are spinning at a low speed, then the large molecules in the cell separate out first. For example, whole cells, nuclei, cytoskeleton, these separate out. When you are spinning at an even higher speed, then the smaller ones like mitochondria, chloroplast, lysosome and peroxisome at an even higher speed and longer duration, Then you get even the smaller ones such as microsomes and small vesicles. Even higher you get ribosomes and viruses. So this is how you can perform differential centrifugation to separate out mixtures, separate out the components of a particular cell. Okay. Right. Now try to answer these questions. Question. Which among the following statements is or are correct? One is soluble proteins have a higher density and lower sedimentation coefficient. Nuclei have lower density and higher sedimentation coefficient. RNAs have lower density and higher sedimentation coefficient. Mitochondria has higher density and lower sedimentation coefficient. So if it has higher density, it is going to separate out first. Right? That is what differential centrifugation says. B. Okay, let's see. So see, sedimentation coefficient depends upon a lot of different factors. Okay, so it depends upon the centrifugal force, the particle that is being separated, and the properties of the media. And that is why you need to take all of this into consideration. So yes, D is the right answer. Nuclei have higher density and lower sedimentation coefficient. So all the different terms and their explanations are given and you can refer to these. Okay. Now we go to something simpler. So this is a question on protein purification, and the systematic diagram shown below represents the construction of recombinant DNA to purify a protein of interest. Okay, so A and B together you get C, and from there you get D. So A, B, C and D, what are they? So recombinant plasmid DNA expression vector, and then DNA ligase, and then cloned DNA. Right, B. Okay, let's see. So B is DNA to be cloned, expression vector, DNA ligase, recombinant DNA plasmid. Okay, so all of you are saying that it is B. Okay. Yes, that's correct. It is B, because see, your insert and the vector, take both of these, you ligate them and you get the recombinant DNA plasmid. Right. So in the process of construction of recombinant DNA to purify protein of interest, the expression vector, DNA and the DNA which is to be cloned combined with each other with the help of DNA ligase protein to form the recombinant DNA plasmid. Okay. Now, the next one is which of the, okay, we have a lot of questions left. Okay, okay, we'll go quickly and finish them off. Right. So question on protein purification. Which of the following is an example of prokaryotic expression vector? Yeast, virus, bacteria, mammalian cell. Choose which is prokaryotic expression vector. This should have been prokaryotic expression system. Yes, bacteria. That is the correct option, because yeast and mammalian cell, these are not prokaryotic, and virus is not a host. Okay, so bacteria is the only prokaryotic. Okay. So they are asking about vector here. Okay, the others are eukaryotic, and virus, it is used for cloning also. It is actually used as vector also. Right. Now we will go for the next one. So IPTG is added into bacterial culture media to induce the protein expression. Now, which of the following is not true about IPTG? 1. It is a molecular mimic of allolactose. It is a part of the metabolic pathway. It triggers transcription of lactoperon. So, which of the following is not true? This you guys know. Try to answer. B. Okay. No, the right answer is 2 only, because 1 and 3 are correct. Right. So IPTG is a molecular mimic of allolactose, and IPTG triggers the transcription of lactoperon, because only when IPTG is induced your repressor cannot come and bind to operator anymore. That is why the polymerase enzyme, it comes and sits on the promoter site, and then the transcription of the downstream genes begin. So therefore it triggers transcription of the lactoperon. That is correct. But then it is a part of a metabolic pathway. That is not correct, because IPTG is not metabolizable. Right? Okay. Now see, this is explained here, explained better. So when the OD is around 0.7, then you are adding IPTG, and what happens is the lactoperon is this repressor is no more able to bind to lactoperon, and that is why you are getting your protein of interest synthesized. Now, what is autoinduction? So autoinduction means when the concentration of the glucose in the media decreases, then what happens? Lactose gets converted into allolactose, and then this process starts. So when there is both glucose and lactose in the media, then autoinduction can even happen. Okay. Now, question on protein purification. In size exclusion chromatography, the column matrix and solvent are selected to dash absorption, and elution is done using dash buffer throughout the experiment. So, adsorption in size exclusion chromatography, do you want to minimize it? Do you want to maximize it? And elution is done using same buffer or different buffer throughout the experiment. C. C is maximize and same. Okay, let's see. Okay, so the answer is actually B. In size exclusion chromatography, the column matrix and solvent are selected to minimize the adsorption. You don't want it to adsorb to stick to the column, because you want to make sure that it is moving when it should travel through the pores, right? In affinity chromatography, it is supposed to stick to it. In ion exchange chromatography, it is supposed to stick to the resin. In these two, it is supposed to stick to the resin. But in size exclusion chromatography, it is supposed to move through the pores and elute. And yes, it is the same buffer. That is correct. Okay. Right. Now we'll go to the next one. So in ion exchange chromatography, a protein can be eluted from a matrix using dash salt concentrations that dash the affinity of the protein for the matrix. Tell me, this is also something that you know. So in ion exchange chromatography, what is going to happen? Do you want to increase the salt concentration in the buffer or decrease it? And what do you want to do with the affinity of the protein for the matrix? For elution, not flow through. So elution means it is the later stage, right? What do you want to do? C, okay. Higher and reduces, that is correct. That is correct, because initially for flow through, your protein of interest is supposed to stick to the column, right? And you want to increase the affinity, and you are using low salt buffer. But then for elution, you are using high salt buffer, and you are reducing the affinity so that it can elute. It can come out of the column. Correct? Higher and reduces. Right. So now, which among the following is false about HPLC? The sample moves through a column containing stationary phase under high pressure. High pressure limits diffusion and increases interaction with the chromatography media. It gives very high resolution of the components. It separates mixtures into compounds according to their molecular weight. Very straightforward. So one of the options is about the name of the HPLC process. Okay, you're saying that it is B. Right, let's see. So the right answer is actually D. Why is that so? Because the sample moves through the column containing stationary phase under very high pressure. That is true because it's high pressure liquid chromatography, high performance liquid chromatography or high pressure liquid chromatography. So that is already there in the name. Now second one is high pressure limits diffusion and increases interaction with the chromatography media. The third one says that it gives very high resolution, which is 100% true. Now the fourth one, it is saying that it separates the compounds according to their molecular weight. But what HPLC does is, we saw here in this slide that HPLC is actually separating the components based on their affinity with the matrix, right? So the desired analytes, so see, analytes with higher hydrophobicity will interact more strongly with the stationary phase and will take longer time to elute from the column. So that means that HPLC is separating the components based on their binding property with the resin. Okay, now we'll go to the next one. Okay, so we'll go on to the next question. So next question says, which of the following statements is or are correct? In dialysis, molecules diffuse through the, so what is dialysis? I did not talk about dialysis, did I? So dialysis is a technique where there is a bag. It's a dialysis bag. It is semi-permeable. So that means solvent molecules can pass through it, but the bigger molecules are retained. It is used for buffer exchange. You can also do buffer exchange using size exclusion chromatography. Okay. So see, in dialysis, molecules diffuse to the semi-permeable membrane if smaller than pore size. Dialysis is used to remove low molecular weight compounds. In ultrafiltration, molecules migrate through semi-permeable membrane under pressure. Ultrafiltration can be used for crude size fractionation and buffer exchange. So which of these do you think are correct? A. Okay. Yes, that's correct. A is the right answer. Okay. Now we go to the next one. So we were talking about that formula, if you remember, partition coefficient, right? So the formula is given there, and now they are asking what is incorrect about these statements about gel filtration chromatography. So particle size determines the void volume. Vt, that is total column volume, minus V naught, which is the void volume, is the volume occupied by the gel space including the matrix. Partition coefficient, that is KAB, is inversely proportional to the log of molecular weight. Partition coefficient is equal to this, where BE is the elution volume. So you need to remember the formula. Okay, now which one do you think is incorrect? How are you going to relate this log of molecular weight with volume of elution? It's this, a negative slope. So if this is the graph, This is your y-axis, which is log of molecular weight. Your x-axis is volume of elution. Then this is the graph with negative slope. Now that means the volume of elution is inversely proportional to the log of molecular weight. Then how are you going to relate? Okay, so you are saying that it is A. Let's see. Okay, so C is the incorrect statement. And particle size determines the void volume V naught. So V naught is determined by the size of the beads, right? So if the bead is relatively smaller, then the void volume will be more. If the bead is larger, then the void volume will be less. Okay. So see, partition coefficient KAB is directly proportional to the molecular weight in the resolving range. Okay. Now the next one is in the first step in two-dimensional gel electrophoresis. So what happens in two-dimensional gel electrophoresis is that there is an isoelectric focusing. That means first the gel is there and there are like proteins with different pI and they are traveling. Now at different pH they are going to stop and precipitate. So that is this separation, horizontal separation, and then the current is flown this way and the proteins are separated based on size like normal pH. So this is one dimensional separation and second dimensional separation. That is a 2D gel. Okay. So in the first step in two-dimensional gel electrophoresis generates a series of protein bands by isoelectric focusing. In the second step, the strip of this gel is turned 90 degree and placed on another gel containing SDS and the electric current is again applied. So in this second step, the individual bands become visualized by interacting with protein-specific antibodies in the second gel. Proteins with similar isoelectric points become further separated according to their molecular weight. And the individual bands become stained so that the isoelectric focus patterns can be visualized. The proteins in the bands separate more completely because the second electric current is the opposite polarity to the first. So what happens in the 2D gel? Tell me. B. Okay. Yes, that is correct. So how does a 2D gel look like? See, this is a strip of gel. Initially, they are going this way. Then they will go this way. So initially they are separated based on their PI. Now when the proteins, two or more proteins have the same PI, they will migrate along the same line, but they will separate out due to difference in their molecular weight. So the proteins with similar isoelectric point become further separated according to their molecular weights. In the second step of the 2D gel electrophoresis, proteins with similar isoelectric points become further separated according to their molecular weights. Now why are they talking about this 90-degree turning of the gel? Because initially the current was flowing in this direction. Now the current is flowing in this direction. So that is a 90-degree difference, right? Okay. Next one is gel filtration chromatography separates proteins based on their PI, secondary structure content, morphology, effective size. This you people know. Simple and direct question. Tell me. Chalo, this is simple. So right answer is definitely D. Okay. So why is it D? Because always remember size exclusion chromatography, separation based on size. SDS page, separation based on molecular weight or size. Right? So this is very important. It can even come as match the following. So don't confuse with that. Secondary structure content is CD spectroscopy. That tells you about the secondary structure content. Okay. Right. Now we will, okay. So these are the different references. You can refer to these different contents. Okay. And I think I have included this. Okay. So I'll also include this virtual labs program links. So I think this was there in one of your slides, possibly the first one or the second one. So I'll include that too. It will be easier for you to learn from there as well. So all of these different techniques are there: size exclusion chromatography, affinity chromatography, and then SDS page. All of these different techniques are there from this virtual lab courses. So there you can, those simulations and everything are there. So you can actually refer to those and try to understand how in practical these experiments work. So there they'll ask you to press a button and then a buffer will be poured into this SDS page container and all that till you make the gel and all that. Okay, so affinity chromatography is there, gel extraction chromatography, gel filtration chromatography, all of these things are there. So you can refer to that. I'll include that. And besides that, these are the different things that you can refer to for protein isolation and purification. Okay. And in the last batch, the students had asked me to include free sources from which they can practice exam questions. So I have not removed these slides. So this should be useful for you guys as well. And you can go through these. Okay, these are all different free sources which help in solving your doubts and solving your problems very well. Okay, thank you so much. Thank you for your time. So we'll end the session here. If you have any doubts, you can ask me. If I'm not able to answer today, I'll answer to you in the next session. Thank you so much. Thank you. Okay, so let's end the session today. Thank you so much to everyone for your time and your response. Okay.